Hi, it's Ademide, Ellie, Maya, Chloe, and Ben, and this is our podcast on the animal models of asthma. The learning outcomes that we are going to look at today are: what is asthma, the principles of validity, and the three models that we're looking at today are the mouse model, guinea pig model, and the primate model. We then also look at alternatives to the animal models. We then conclude with the ethical dilemmas that we face. What is asthma? Asthma is a long-term respiratory condition marked by spasms of the bronchi in the lungs, causing breathing difficulties. It is usually connected to forms of hypersensitivity. In the UK, around 5.4 million people are currently receiving treatments for asthma. That is 1 in 12 adults and 1 in 11 children. In the US, it causes nearly 2 million emergency room visits per annum, with 250,000 annual deaths worldwide. Asthma affects the airways by causing the muscles around the walls of the airways to tighten so that the airway becomes narrower. The lining of the airways can also become inflamed and start to swell. This inflammation is thought to contribute greatly to the long term damage that asthma can cause to the lungs. There is sticky mucus that is sometimes produced and can build up in the airway, which can cause obstruction and difficulty breathing. Fortunately, these changes are short term and they can be reversed using medication. The symptoms of asthma are well known, and they are tightness of chest, wheezing, shortness of breath, and coughing. The most popular treatment for asthma is a short acting beta 2 adrenoceptor agonist known as salbutamol, which is the first line of treatment for the disease. Salbutamol stimulates beta 2 adrenergic receptors, which are predominant receptors in the bronchial muscles in the lungs. Activation of the receptor leads to cyclic AMP stimulation that induces relaxation of the muscles all the way from the trachea to the terminal bronchioles.
The principles of validity judge whether the animal models are effective. Construct validity is an assessment of how closely the origin of the animal model mirrors the origins of the disease in man. Face validity assesses the extent to which the phenotype of the animal model matches the phenotype of the human disorder. Predictive validity is the ability of an animal model to predict whether a drug will be active against the target disorder in man. We'll now look at mice as models for asthma. Animal models are necessary as they mimic pathophysiology of disease, meaning we can discover new mediators and other contributing factors. They also allow us to evaluate safety and efficacy of therapies to avoid wasting time on clinical trials, as well as avoiding harmful side effects on volunteers. Also, there are many ethical requirements to conduct human studies, so if these aren't met, an animal model is a suitable replacement. Mice are the most common animal used to model asthma. The most common strain is BALBC, and the inflammatory response is triggered by a valbumin with aluminium salts. The key advantages to using the BALB-C mice are that they are cheap, easy to handle, easily sensitised to the disease, and the reagents used are largely available. However, there are some disadvantages. The lung anatomy of a mouse varies greatly to that of a human, so the results are not directly transferable. The distribution of lung inflammation also differs to that of human asthma. There is a lack of chronicity of the response to the allergen. Therefore, it's difficult to measure how similar the process is in chronic asthma in humans. Finally, a tolerance develops after repeated exposure to the allergen. Symptoms that correlated with asthma included airway inflammation, epithelial hypertrophy, goblet cell hyperplasia, and airway hyperresponsiveness. Both acute models and chronic models have been used to identify mediators of asthma. In the acute models, it was found that there were high serum levels of immunoglobulin E. Interleukin 13 and 5 were also found to be key mediators in the inflammatory response in the airways. Interleukin 13 and 5 showed to only be functional in eosinophilic airways, so couldn't be applied to all aspects of asthma. However, successful findings from Bright-Bill et al. Found blocking IgE was highly effective treatment in these mouse models. Clinical trials and further development lead to the production of omalizumab. Omalizumab is a drug example that has been discovered through the use of these mouse models. It inhibits the binding of IgE on mast cells, which prevent the inflammatory response in airways.
Guinea pigs are among the oldest models of allergic airway responses, and studies on them have been done for over a century. They can be used in asthma studies due to their airways being sensitive to allergens. Unfortunately, working with guinea pigs can be quite challenging. Guinea pigs exhibit pronounced airway hyperreactivity and heightened sensitivity to anaphylactic shock. Also, the cost of the animals, reagents, and the limited availability of specialized instrumentation further limit their use as a research model. There are also not many different strains of guinea pigs, so comparative studies are difficult. Guinea pigs offer many advantages in preclinical studies relevant to asthma. The airway response to allergen challenge is very similar to that of the human airways and very different to that of the mouse. Receptor pharmacology in guinea pigs closely resembles that of the human airway, more than other animals. There are very few exceptions of metabotropic and ionotropic receptors that do not follow these similarities. Complex regulators of cell function related to the neuronal and mast cell activation, second messenger formation and metabolism, and cytokine gene expression are nearly identical in the two species. The autonomic innovation of the guinea pig airways and responses to experimental interventions are also very similar to that in humans. Compared to rodent models of asthma, guinea pigs are readily sensitized to evalbumin. Evalbumin is a frequently used allergen that induces a robust allergic pulmonary inflammation in laboratory animals. Work in the 1960s on the lungs of guinea pigs led to a greater understanding of the function of chemicals that are released by tissues when normal biological processes are disturbed. In 1979, substances called leukotrienes were discovered to be the principal cause of the symptoms of allergic and inflammatory conditions. Many fundamental processes, mediators, and regulators of airways disease pathogenesis were discovered or demonstrated first in guinea pigs. including the Schultz-Dale reaction, the actions of histamine, beta adrenoreceptor subtypes, thromboxane, etc. The inhaled medications that are the backbones of asthma treatment were developed using guinea pigs. Studies conducted using the guinea pig were enormously beneficial in selecting reliever drugs that had the right properties that enabled asthmatics to take these medications twice daily to provide them protection against the symptoms of asthma during the day and also during the night. Animal experiments in guinea pigs and then primates led to the development of leukotriene receptor antagonists. These drugs, introduced in 1998, were the first new type of treatment of asthma in 20 years. The advantage of this new type of therapy is that they can be taken by mouth. These drugs are effective on all types of asthma. Guinea pigs can provide models for asthma with high validity in all aspects. This is due to the underlying mechanisms and symptoms being the same as in the human form of asthma. Predictive validity has been shown to be sufficient in many cases in the past.
Asthma models in primates. The rationale for development of asthma models in primates is centered around their similarity to humans both genetically and physiologically. The lungs of primates are particularly similar to the ones of humans in terms of their anatomy, histology, ultrastructure, and sound. These similarities allow the same pulmonary function used in human studies to be conducted in primates with similar techniques and instruments. Physiologically and immunologically, both humans and primates respond to allergen challenge in similar ways. When cohorts of sensitized primates have been repeatedly challenged to allergens, they have shown a variety of characteristics which can also be observed in chronic asthma human sufferers, with only episodic wheezing and airflow obstruction being a feature that cannot be observed in monkey asthma models but can be observed in human atopic asthma. Rhesus monkeys were first sensitized to the Ascaris parasite 40 years ago and later on sensitized to human allergens such as house dust mites. Exposure to these compounds was seen to trigger T-cell memory responses and increase immunoglobulin E levels. Non-invasive forced oscillation techniques have now been used to measure changes in pulmonary function in these monkeys after being exposed to these allergens. And physiologically, the rhesus monkeys underwent airway challenge, which was followed by airway eosinophilia and hyperresponsiveness. Currently, no drugs have been developed solely based off of asthma models in only primates. Instead, primates have been used in the preclinical evaluation of asthma therapies.
Alternative methods to animal models. Firstly, why are we looking for alternative models? In short, if there is a way of conducting a test without the involvement and therefore potential pain and suffering of an animal, there is an ethical obligation to choose the non-animal route. An advocate for this approach is the National Centre for the Replacement, Refinement and Reduction of Animals in Research. This body wants us to focus on the 3 R's within its name and use these stimuli as a way of avoiding pain and suffering that an animal may be subjected to in animal testing. There are 3 alternative models to animal testing: human tissue and tissue engineering approaches, which may appear to be the most plausible method for asthma research, using non-mammalian model systems as it's thought that the pain and stress felt by these types of organisms is considerably less than their mammalian counterparts. And finally, applying mathematical theory to certain aspects of asthma's mechanisms. Human tissue and tissue engineering approaches. This is the most feasible approach to accomplishing the previously discussed 3Rs. This will allow the study of asthma in human tissue, which benefits understanding greatly, whilst reducing the numbers of animals involved in animal testing. Obtaining and testing on human tissue can have its difficulties, so the National Centre for the Replacement, Refinement and Reduction of Animals in Research have set out key steps that will enable this shift from animal to human tissue studies. The steps. Workshops will enable greater vocalization and discussion on using human tissues, which in turn allows for the improvement of procedure and testing on a national scale. The interaction with journal editors will allow more studies that use human tissues to be published, allowing for the normalization of human tissue studies and hopefully from this inspire the use of more human tissues. And finally, there should be more networked access to human tissues. Since some areas/institutions can have greater amounts of tissue available than others, there should be a system by which the human tissue resources are pooled together and then nationally redistributed evenly. Using non-mammalian model systems. The use of non-mammals in respiratory research is almost zero, since it is hardly feasible in these types of animals. Although there is some possibility that there may be an aid to understanding the molecular mechanisms of the disease, there is very little research in this area. This is why the British Thoracic Society Difficult Asthma Network have hosted a workshop to explore the feasibility of using non-mammalian models to provide pathways and potential approaches for studying severe asthma. The result of the workshop led to a strategic call to invest £500,000 into research in various non-mammals. Applying mathematical modelling. These models offer new tools to better analyse existing data and inform the future experimental approaches with reduced reliance on animal models. They have been particularly effective for specific areas, notably examining steroid responsiveness in COPD and asthma patients, along with studying the smooth muscle cell proliferation and apoptosis.
So, what are the ethical problems that we face when using animal models? There's an argument that we shouldn't use animal models because it doesn't take into account the animal's rights. But the defense against that is that by studying animal models, it is possible to obtain information that cannot be learned in any other way. When a new drug or surgical technique is developed, it is deemed unethical to test the drug or technique on human beings because of unknown side effects. Animal testing is seen as the lesser evil of the two, to make sure that the drug or technique is safe and effective. Animal models offer experiment models that will be impossible to replicate in humans, as animals' diets and reproduction and environment are tightly controlled to avoid variance in the results. So it's harder to find a way to do that in humans than in animals. It can also be argued that fewer animals are used in research compared to animals kept as pets and used for food. There is a 96 million animal difference. So now you have heard from Ademide, Ellie, Maya, Chloe, and Ben, and that is the conclusion of our podcast on the animal models used in asthma research. We hope you learned something today. Thank you. Bye bye.